Hi, this is Malinki. Welcome to my channel, Voice of Malinki. Hope you all are safe and doing well in this pandemic situation. COVID-19 has ruined our life and changed the world, and it is also a very hot topic in today's biology. So if you are a life science student preparing for CSIR NET, GATE, ICMR, DBT, IISc, TIFR, JAM or any other PhD entrance test, or you're in your graduation or post-graduation and willing to join a reputed biotech company, this video is for you. Because it is your duty to know the basics of COVID-19. Questions will be asked to you regarding this in your next interview. So be prepared for that. If you have good rate in your semester and you are specialized in the field of botany, zoology, biotechnology or microbiology, but you don't know the basics of SARS-CoV-2, you are not going to crack the next interview. And it's a shame for a biology student to not to know the anything about this disease. And if you are interested in a molecular biology or virology laboratory, you will be definitely asked these questions. So I decided to make some lectures on this topic to make your life easier, and the lectures will be very much simple so that you can understand them clearly no matter what biology background you have. I have split the whole stuff into few lectures because if I say everything in a single video, it will be too much for you to take and too much for me to deliver. So today I will discuss about the introduction, structure, taxonomy, epidemiology and pathophysiology of COVID-19. This is the lecture 1 of this series, and if you are new in my channel, please don't forget to subscribe my channel, and if you like my video, please do like, comment and share my video. So the first thing is coronavirus is not a very new virus. So it has infected us already twice before this 2019 pandemic. In 2002, coronavirus has infected us, and that time the disease was named as SARS, that is Severe Acute Respiratory Syndrome, and the virus was called as SARS-CoV-1, that is SARS coronavirus 1. One, because this virus was the first identified strain of SARS coronavirus, and the first case of this disease was found in China. Now, what is case fatality rate or CA4? So the case fatality rate or CA4 is the number of deaths divided by total number of cases into 100. So, in this particular disease, the total number of cases was 8,000. That is, 8,000 people were infected by this virus. And among those 8,000 people, 800 people died. So, 800 divided by 8,000 into 100 yields 10%. So, the case fatality rate was 10%. The virus has spread to 29 countries. That's why I am calling it a mini pandemic. Now, if we consider the route of transmission, bat is the primary host that was infected by the virus. In the bat, the virus was mutated and then it was transmitted to an intermediate host, that is a civet. Civet is a kind of cat. In civet's body, the virus was mutated again and then it was transmitted to a human. Maybe you are laughing at this picture I have drawn here. It's more like penguin, but please consider it as human. And the virus causes fever, pneumonia and acute respiratory distress syndrome or ARDS. Now, after 10 years of the SARS-CoV-1 pandemic in 2012, coronavirus again attacked us. This time, the case was first identified in Saudi Arabia and the disease was named as MERS. that is Middle East Respiratory Syndrome. That is named so because the viral infected persons were mainly from Middle East and the virus was called as SARS-CoV or SARS coronavirus. This time, the total number of infected persons was 2,500. Among them, 850 persons died. So, 850 divided by 2500 into 100 will yield 34%. So, this time, the case fatality rate or CFR was 34%. That is pretty much higher and it's dangerous. The virus has spread to 27 countries and causes a mini pandemic again. If you consider the route of transmission, bat is the primary host again. So the virus first attacked bat where it was mutated and it was transmitted to an intermediate host that is camel. So in case of SARS-CoV-1, the intermediate host was civet. In case of SARS, it is camel. Although the primary host is same, that is bat for both the cases. Now in the body of camel, the virus was again mutated and it was transmitted to a human body. Symptoms are quite same as SARS-CoV-1 like fever, pneumonia and acute respiratory distress syndrome or ARDS. Now, for the third time, this coronavirus has infected us again in 2019 November. This time, the disease is named as COVID-19, that is coronavirus disease 2019. And the virus is called as SARS-CoV-2 or SARS coronavirus 2. And 2 indicates the second identified strain of SARS coronavirus. First strain attacked us in 2002 and the second strain has attacked us this time. And like 2002 pandemic, again the first identified case was found in China. And this time the case fatality rate is only 3%. I haven't put the data over here because the number of infection and the number of death is getting changed every day. And if I put it today's data and you watch my video tomorrow, just tomorrow, 1 or 2 months later, you cannot relate. But till now, the case fatality rate or CFR is only 3%. I am saying only. Yes, I am saying only because it is pretty much lower as compared to the previous two SARS-CoV-1 and SARS. It was 10% in case of SARS-CoV-1 and 34% in case of SARS. Then why we are scared? We are scared because although the death rate is low, but the disease is very much infectious. In case of SARS-CoV-1 and SARS, very few people were infected. But in the current pandemic, millions of people are already infected and the number is increasing day by day. So it's scary in that way. And if you consider the route of transmission, again bat is the primary host. So bat is common primary host in all these diseases. In the body of bat, this virus is mutated and it is transferred to the intermediate host, that is pangolin. In the body of pangolin, the virus is again mutated and gets transmitted to a human body. Symptoms include fever, cough, chest tightness, difficulty breathing and shortness of breath, that is also called dyspnea. So if you study an infectious disease, you have to understand two things. One is R0 or R0 and another is SI. R0 is the reproduction number. So it is the number of infections spread by one person. Or you can say how many persons are infected by a single person at a time. It indicates how infectious a disease is. So let's explain it here. Suppose this person is having COVID-19. He is unaware of his condition and moving here and there. He is coughing, sneezing and releasing respiratory droplets. Now, these 6 persons are around this person and they are infected. These 6 persons are now infected. So one person is capable of spreading the virus or spreading the infection among 6 persons. Now each of these infected persons are capable of spreading the infection among 6 more persons. So imagine how many persons are infected just from few people. So R naught of COVID is 5.7. That is, one person can infect 5 to 6 persons at a time. In case of common flu, R naught is 1.3. So a single person is infecting another person. He is infecting one more person. So spreadability is less in case of flu, right? Which is very high in COVID-19. That makes the disease dangerous, and that's why we are advised to keep ourselves quarantined. The next thing you need to know is serial interval or SI. SI is the gap between the onset of symptoms in person A and person B who is infected by person A. Let's explain it here. Suppose this is person A. At this point, he is infected by the virus. But he doesn't know that he is infected because he doesn't have any symptoms at that time. When we get infected by the virus, we don't show any symptoms on that very day. The virus needs to get replicated in our body to show some more symptoms and make us sick. It takes time for them. This time varies and depends on the viral strain and the viral type. So, person A is infected by the virus and leaves his room. He is going to market, a mall, a hospital, etc. Now, person B, this person is infected, it's getting infected by person A at this point. So, at this point, he is getting infected. And at this point, person A will show some symptoms. So, A will show some symptoms here. But he has already infected person B. And person B will show some symptoms here. So the gap between these and these is the serial interval. More the SI, opportunity for virus transmission is also more. Less the SI, opportunity for virus transmission is also less. Because if the SI is lower, you are aware and you know you can isolate yourself faster. That will reduce the chance to infect some other persons. And if the SI is larger, you will take more time to know about the disease, and by that time you will already spread the disease. For COVID-19, the SI is very high. It is 4.2 to 7.5. For common flu, SI is lower. It is just 3. This makes COVID-19 even more dangerous. Now I will explain the taxonomy of it. Coronaviridae family is divided into 4 genera. Alpha coronavirus, beta coronavirus, gamma coronavirus and delta coronavirus. Among them, alpha and beta coronavirus infect humans and gamma and delta coronavirus infects bat. And beta coronavirus actually infects us to cause SARS-CoV, SARS-CoV-2 and MERS-CoV. So, whenever gamma and delta coronavirus infects a bat, it has to be mutated in the beta form before infecting human. So, in this way, the coronavirus is mutated and it is infecting us. Now let's talk about the structure of coronavirus. So it has a lipid bilayer, this green one. This is the lipid bilayer. 70% alcohol or hand wash can break down that lipid bilayer. That's why we are advised to wash our hands many times, because washing can break down these lipid bilayer and kill the virus. And some proteins are embedded in this bilayer. These are spike protein or S protein, envelope protein or E protein, membrane protein or M protein. So these are the proteins that are embedded in this bilayer. We need to know about S protein or spike protein only, because this is the protein that binds the host cell surface. So this is the entry point. If a virus doesn't have this S protein, it cannot infect us. We don't need to know about the other proteins here. They are all structural proteins. The genomic material of the virus is RNA, and this is a positive sense single-stranded RNA. The size of this RNA is 27 to 32 kb. It has a 5' cap and 3' polyadenylation tail. And nucleocapsid proteins, this red one, these are some nucleocapsid proteins. So these proteins bind this RNA. Now, if you know what is positive sense RNA, then it is okay. But if you don't know what is positive sense RNA, I am explaining it here very quickly. So, positive sense RNA actually acts like an mRNA. It can translate proteins directly. That's why it is called as positive sense, because it can sense, and that makes a virus more dangerous, because when a virus possesses a positive sense RNA, it will enter into the cell and directly gets translated. So it saves its timing and replicates very fast, because it doesn't rely on replication or transcription, etc. That makes the COVID-19 more efficient. Now, all these structural proteins are well conserved except for the spike proteins, and that showed a high rate of mutation in COVID-19. So, the envelope protein, membrane protein, nucleocapsid protein, they all are conserved in the cases of SARS-CoV-1, MERS-CoV and SARS-CoV-2. But the spike protein is actually mutated in COVID-19 and makes it novel. Okay, so before explaining the pathophysiology, I will tell something about surface tension and surfactant. Without the knowledge of these surface tension and surfactant, you can't understand how the coronavirus works in our body, or how does our body respond during the infection. If you are a physiology student, you already know about this, but if you are not, you can understand now. So tendency to shrink into the minimum surface area possible is known as surface tension. It occurs in the air-fluid interface. So these are some healthy cells. Now due to the surface tension, these cells will shrink like this, and lung cells mainly experience more of these, because lung is full of air. We inhale and lung gets filled with air. So the cells are in the contact of air, and we know cells are filled with fluid. So a fluid air interface occurs that makes the cells to shrink. Now lung cells are specialized for gas exchange. When cells shrink like these, they cannot exchange gas properly. Now some cells of lung secrete surfactant. So these are type 2 pneumocytes who secrete surfactant. The role of surfactant is to reduce the surface tension so that cells get their proper shape and play a role in proper gas exchange. So this is the stuff you need to know. Now I will explain the pathophysiology. So this person is having COVID-19 and he is not keeping himself isolated. He is going out of his home. Now he is sneezing, coughing and releasing respiratory droplets in the air. whatever the things are there in that place, like table, chair, bed, almirah, table lamp, etc., everything will be contaminated by those respiratory droplets. Right? Now this person is innocent and doesn't know about the infection. He is now touching those contaminated objects and touching his nose or mouth afterwards, or inhaling the virus directly from the air. So he will get infected now. The virus will enter through nose or mouth and travels through respiratory tract and enters lung. Now, in lung, type 2 pneumocytes are there. This is the cell, this is the cell, this is the type 2 pneumocyte. So, in lung, these type 2 pneumocytes are there. As I have already talked about them in the previous slide. Now, these type 2 pneumocytes are the target of coronavirus. Why? These cells have ACE2 receptors, that is angiotensin converting enzyme 2. These ACE2 receptors are found in the cell membrane of these cells. Now, ACE2 receptors are special types of receptors found only in these cells and enterocytes. The ACE protein or spike protein of virus has the high affinity for these ACE2 receptors. So viral ACE proteins will bind these ACE2 receptors. Now, once binding occurs, the virus gets entered in the cell cytoplasm through the process of endocytosis. When virus get endocytosed into these cells, lysosome will break down the lipid bilayer and viral RNA will come out of it. So the naked viral RNA is now in the cytoplasm. Once the viral RNA is there in our cytoplasm, it will do two things. The RNA is very clever. First of all, it will synthesize lots of RNA. But how? Well, it will take the help of our host cell RNA-dependent RNA polymerases. These are some enzymes that generate RNA from a RNA template. That's why the name is RNA-dependent RNA polymerase. So it will generate numbers of viral RNA from the RNA template itself in the cell. Now, one more thing these viral RNA will do. It binds our host cell ribosome. So our cell cytoplasm possesses numbers of ribosomes because continuously translation process is going on. Now these ribosomes will be hijacked by the viral mRNA or viral RNA. Binding of the ribosome and the viral RNA will generate proteins. Because translation is very easy here as these are positive sense RNA and directly produces viral proteins in the cell. Now these proteins include membrane protein, envelope protein, nucleocapsid protein, spike proteins, etc. So all the structural proteins of the virus will be made. Now these proteins and RNA that are already formed will combine to generate new viruses. So new viruses are now formed and they cleave the host cell membrane and come out of cell. So in this way, type 2 pneumocytes are destroyed because the plasma membrane are now cleaved. When these cells die, they release specific inflammatory mediators. Now, these inflammatory mediators will stimulate macrophages. These macrophages will now release some specific cytokines like interleukin-1, interleukin-6, tissue necrosis factor or TNF-alpha, etc. These cytokines will induce fever. Now, these cytokines will not only induce fever, but they will enter the bloodstream and cause dilation and increase capillary permeability. Dilation means the blood vessels get wider and Increased capillary permeability will allow some molecules to come out and in the bloodstream. As capillary permeability increases, plasma from the bloodstream will leak into these interstitial spaces. Interstitial space is the space between the tissue and bloodstream. Okay, so now plasma comes out into the interstitial space. As it comes out, it will cause tissue swelling, that is scientifically termed as alveolar edema. Any type of swelling is called as edema, and because this is lung tissue, we are saying it alveolar edema. Now this excess fluid accumulation around the lung cell will generate pressure that will impair gas exchange, that may cause respiratory failure, because lung cells are specialized in gas exchange and they are not able to do that. That causes respiratory failure. And whenever the type 2 pneumocytes are destroyed, they are not able to produce surfactant anymore. That in turn increases surface tension and alveolar collapse. And apart from the plasma leakage, this capillary permeability will also allow some neutrophil influx into lung. Neutrophils will actually release ROS or reactive oxygen species and proteases in order to destroy the virus. But these ROS and proteases are not cell-specific. They will also cause mass destruction of all alveolar cells, including type 1, type 2 pneumocytes, etc. Now, type 1 pneumocytes have an integral role of gas exchange, so that will be damaged. Now, type 1, type 2 pneumocytes and other cells all are dead. Dead cells will generate cellular debris that will lead to consolidation, or in a simple form, you can say they will block the tissue, they will jam the tissue. So when tissue is blocked, again the gas exchange will not take place. That will cause low level of oxygen in blood or hypoxemia. When lung tissue is consolidated, you cannot breathe properly. You cannot take a long breath. The situation is named as dyspnea or shortness of breath. And tissue blockage will not allow to take a deep breath. So you will cough. Here I have already mentioned that dilation occurs due to these cytokines. That means blood pressure will drop as the blood vessels are widened now. So blood pressure will drop. This will cause hypotension. Now hypotension will cause decreased perfusion. So perfusion is the delivery of blood to a tissue. Now blood provides all the necessary elements like food, mineral, oxygen, etc. to tissue. So when blood pressure is low, these materials could not be delivered to the tissue properly. And all the tissues are dependent on blood for the basic stuff. So when blood delivery is hampered, organs start not to function properly. This causes multi-system organ failure or MSOF. If multi-system organ failure occurs, we cannot survive anymore. And apart from the type 2 pneumocytes, ECE2 receptors are also present in enterocytes. Enterocytes are the intestinal cells. That's why COVID-19 can also infect those cells, and patients with COVID-19 may also present some distinct gastrointestinal symptoms. Hope you understood this concept. Thank you and bye-bye.